Welcome to the Phelan-McDermid Syndrome podcast, sharing research, progress, and hope. We have a real treat today with two separate interviews looking at really unique, novel pathways in Phelan-McDermid syndrome. First, we have Dr. Boaz Barak from Tel Aviv University, where he's an associate professor studying myelination in SHANK3. In addition, we have Dr. Haitham Amal from Hebrew University of Jerusalem, who is also a visiting professor at Harvard University and Boston Children's Hospital, who is studying nitric oxide in SHANK3. These are two really innovative researchers who are doing some great work and offering some potential new therapeutic pathways for our individuals with Phelan-McDermid syndrome. I'm excited for you to listen, and I hope that something good comes out of this in the future. Thanks all. Hi, Phelan-McDermid syndrome families and community. We are so excited today to have Boaz Barak featured today on our podcast to really highlight some new exciting areas in Phelan-McDermid syndrome research. I will let him introduce himself in a second, but I also want to highlight he is a recent award winner of one of the Autism Science Foundation grants as well. And so we're really excited to hear his work today and to introduce a little bit newer of a topic for our families. Welcome.
Thank you, Lauren. So my name is Boaz Barak. I'm an associate professor at Tel Aviv University. I am studying Phelan-McDermid syndrome and Williams syndrome. And I did my postdoctoral training with Professor Guping Feng at MIT, where I got to know this kind of exciting field of Phelan-McDermid syndrome. So that's me. In the last 8 years or so, my lab is studying Phelan-McDermid syndrome.
Wonderful. Well, we're glad that you are part of our community. So the main reason that I really wanted you here, and you were one of the first people I talked to kind of when I started in the CSO position, is you're studying something a little bit different called myelination. And so tell our families kind of what it is you're studying and what your specific project funded by ASF is going to be looking at.
Right. Okay. So as you know, in Phelan-McDermid syndrome, SHANK3 is involved. And SHANK3 is heavily studied in aspects related to those nerve cells, those neurons in the brain, and how they communicate with each other through this structure called synapse. Now, this is heavily studied and there is a nice progress, but our lab is very interested in myelination, which is a layer in the brain or in the body that covers those neurons, those nerve cells that have like a long cable. We call it axons. You actually have it on your paint behind you. So this is like an electrical cable that can help communicate one neuron with another one. And the myelination helps to cover this cable and enables fast transduction of the electrical signal. So if you don't have a proper myelination, the electrical signals from one side of the brain to the other will be affected. And this is something very critical for the brain functionality. If you don't have proper communication, that will be bad for your brain synchronization and so on. And this myelination layer, this myelin, is made by cells that we call oligodendrocytes. And these are critical cells to support and push the electrical activity in the brain. And we discovered that actually SHANK3 is not only critical for neuron-neuron interaction and communication through this kind of synapse, but that actually there is a synapse between this cell that make myelin, oligodendrocyte, and the neuron itself, so that also in this kind of communication, you have a synapse, and SHANK3 actually plays a critical role for the functionality of the oligodendrocytes. So by studying a mouse model with a human mutation, just like in Phelan-McDermid syndrome, we were able to study what are the consequences of this mutation on oligodendrocytes functionality. So this is a whole new field that expose SHANK3 into a new type of cell in the brain that is critical for the activity of it.
Wow, that's fascinating. And I really appreciate your analogy of the electrical plug. And I think hopefully our families who maybe a little bit don't remember their Bio 101 is
Yeah,
that it's really helping with this electrical signal.
right.
Um, and so you found this out about Shank 3 and its vital importance in this role, in this new role. And so what now?
Yeah, so it's not only that we discovered what is affected and what are the deficits on the molecular, cellular, brain circuit, behavior, and so on, we also validated our findings also in human tissue samples, meaning that we took cells from a patient that has the same mutation as we studied in the mouse. So this is a real patient, a human patient that has the Phelan-McDermid syndrome, and by taking cells from this patient and reprogram them into stem cells, we were able to reprogram them now into oligodendrocytes. And then we were able to validate that all of those deficits that we discovered on the mouse level are indeed valid also in human cells. This means that this is clinically relevant. This is something that we can understand that this is also the case in humans and not only in a preclinical study. And further than that, we also wanted to see if we can hopefully improve the condition of myelination. So we try to make a potential genetic treatment, you can say, in which we restored SHANK3 levels in oligodendrocytes that derived from mutant mouse. So now you think about a dish that you have cells inside. Those cells don't express SHANK3 because of the mutation. And now we try to restore SHANK3 in those oligodendrocytes without the SHANK3. So these are cells that derive from postnatal stages, but we try to restore the SHANK3. And we did so, and indeed we were able to restore SHANK3 in oligodendrocytes for the first time in the world. And we saw that restoring SHANK3 was possible. We saw restored SHANK3 protein levels. And by the way, this was a human SHANK3 sequence, meaning that we restored the human genome sequence of SHANK3, which is very clinically relevant. And by restoring SHANK3, we showed that now this restored cell can also present proper proteins that were missing because SHANK3 couldn't hold them. So SHANK3 is like a scaffolding protein. It holds many other proteins in the synapse. And once you restore SHANK3, other proteins were able to be restored, including receptors, which are critical for the functionality of the brain. So this is very exciting. It might be in the future, future cell type, which will be a target for genetic treatment. And that's something that is exciting for the families and for all of us together.
Absolutely. It's really nice to hear sort of maybe this new promising avenue in cellular and genetic therapy. You also mentioned kind of when we were offline earlier that there's also some already available treatments that kind of help at the myelination stage. So can you tell our families a little bit about that too?
Yeah, sure. So my lab is trying to use common drugs that are already FDA approved and that you can just repurpose from one pathology into a new pathology. So by doing basic research as my lab is doing, we can understand mechanistically what is abnormally functioning in the cell. And then if we understand that, we can look at drugs that we know that fix this kind of deficits that we discovered, and we can borrow them from other pathologies. So, for example, there are many myelination deficits pathologies in which, like multiple sclerosis and many others, that myelination is impaired. And there are a whole bunch of drugs that can improve myelination in those pathologies. But now that we understand that also in SHANK3 related pathologies, you have myelination deficits, we now try in my lab two different drugs which are FDA approved, clinically active, safe, but were never tested on Phelan-McDermid syndrome because nobody knew this is an issue. So what I want to say to the families is that by understanding that not neurons only are affected by SHANK3 mutation, but also a new bunch of cells, this might open a new era of testing drugs that can mediate myelination and not necessarily only neuronal aspects, but also myelination. So that's very important and exciting. And hopefully we will be able to have some new findings and exciting things that can be ended up as a clinical trial in which we test the safety and efficacy of these FDA-approved drugs, but now on Phelan-McDermid syndrome.
Right, absolutely. And, I want to highlight two specific points there for our families is that this whole idea of drug repurposing is really important in rare disease because it kind of jumpstarts you in the whole FDA approval process because there are some initial efficacy indications and other disorders or safety ratings, but we still need to make sure it's safe and effective in our individuals with Phelan-McDermid. So it's not something you can, ask your doctor necessarily and get off the counter right now,
Right.
but it brings us a lot closer than if it was just sort of a new small molecule that was discovered or something.
Right. Yeah, I fully agree. I mean, don't think that if you have a drug relevant for myelination deficits, then give it to your kids. No. Let the researchers and the clinicians show that this is relevant, safe, and effective. But the advantages of using repurposed drug is that I don't need to develop a new drug with so many millions of dollars of development and so low chances to really have it effective at the end of the day. And it will take 10 years to develop a new drug, and this is a repurposed drug. So these are great advantages by doing repurposed research.
Absolutely. Well, we will definitely stay tuned to where this is going and how your research is going. I've been, again, particularly excited, I think, because we both have, sort of this repurposed drug avenue, but then also there might be some cellular therapies that also could work. Obviously, those are going to take a little bit more time. And so putting you on the spot a little bit, in 2026, we are having our family conference, which we just announced is going to be in Colorado. Do you think we can expect you to be there and maybe show off your latest findings?
Yeah, sure thing. Count me in. I'll be happy to be there. Yeah.
Awesome.
Exciting.
Well, we're so excited to have you today, Dr. Brock, and I'm glad you were able to share this new exciting avenue for our parents. And like we said, just sort of stay tuned and hopefully we'll have some new exciting findings to share.
Thank you very much, Lauren. Great to be here.
Hi, Phelan-McDermid syndrome community. I am so excited today to have Haitham Amal with us. He is an associate professor at the Hebrew University of Jerusalem. He's also a visiting professor at Harvard University and Boston Children's Hospital, where he's embedded in the Rosamund Stone Zander Translational Center. We are excited to have him today to talk about some of his new developments and research and promising avenues in Phelan-McDermid syndrome. Welcome.
Hi, Lauren. Thank you so much for the invitation and for the PMS Foundation for this kind interview.
Of course. So I'd love to know kind of a little bit about how you got into Phelan-McDermid syndrome SHANK3 work to begin with.
Yeah, so it was a decade ago already, 2015. I joined the lab of Stephen Tenenbaum lab at MIT Biological Engineering, and right away we started a very important collaboration with Professor Goping Fang on the SHANK3 mouse models.
We've definitely had several people from Gopeng Fang's lab on this podcast. He has definitely helped shape a new generation of Phelan-McDermid researchers.
Yeah, Dr. Fang has done a tremendous support and important achievements for the community and for the research field. So we are grateful to have him. Yeah.
Yeah, absolutely. So you were basically the first person, if I'm not wrong, to link oxide to autism spectrum disorder. For our families out there, can you tell us a little bit about what nitric oxide is and what does this even mean?
Yeah, so actually it started at MIT where we found a link, just a correlative link between the nitric oxide and autism. And once I went back to the Hebrew University, we actually started working very hard on proving this link and the causal link between nitric oxide and autism and PMS, of course. So to be, a general statement, nitric oxide is a small molecule, you can call it neurotransmitter in the brain that is important for neuronal and synaptic function and also important for cognition and memory. And we were very lucky to be the first, my lab, to be the first to show that nitric oxide actually plays a crucial role not only on the synaptic and neuronal dysfunction, but also on the ASD or autism plus PMS like phenotypes. And I'm gonna elaborate, of course, on the findings that we had on this regard.
It's not something that probably our families heard much about. So yeah, tell us a little bit more about what your specific findings were and what it means.
Yeah. So actually, when we published the paper 2023 in Advanced Science, I didn't expect this much of attention, in the media and so for now, I think the paper is ranked in the top 5% out of 28 million papers according to the Almetric. And the news was translated into 10 different languages, and it was really exciting. We had, of course, Shank 3 mouse model. We used the 4 to 22 deletion mouse model of Joseph Baxbaum. And we recruited kids, idiopathic kids with autism and also Shank 3 mutation kids. And we received the blood samples of these kids. Plus, we received stem cells samples from a girl with PMS and her mom. And what we found is increased nitric oxide levels in all of these samples. And when we tried our drug, we found reversal on the synaptic and molecular dysfunction. And in the mouse model, we found a reversal of the main or core symptoms of autism, social behavior, communication, repetitive behavior, anxiety, etc. So these are the findings that included more than 1,000 animals, three different mouse models, clinical samples, stem cells, and I think this is why the study was very important for the community and for the scientific field.
Absolutely. And yeah, thinking about 1,000 animals, that's a lot of replication, which is very
Yeah,
important in science.
absolutely.
Is there a way to sort of wheedle down what increases in nitric oxide would do? And like, why is it having those profound impacts on everything from cognition, social communication, anxiety?
Yeah, super, super important question, and also I'll try to use lay term here. And so we in the last 5 years, my lab published more than 20 papers, and most of them included Shank 3 mouse models. And we have many mechanisms that we think that are involved. And the nice thing with nitric oxide is that nitric oxide is a molecule that can affect other pathways. So for example, I think this year or last year, we published paper in Scientific Reports, and we showed the link between nitric oxide and autophagy, which is a very important mechanism in the cell. So this is one mechanism that we think that nitric oxide affect autophagy, and autophagy in a specific regions in the brain affect behavioral dysfunction. The other part that we are now gonna submit the paper, this is for the first time that someone comes and explain why we see increase of mTOR signaling in PMS and in autism. And we, in this paper, found that N-nitric oxide activates mTOR signaling. So once we inhibit NO, we can shut off the mTOR signaling. And this is important because I think there were many failures of the mTOR inhibitors treatment. And I think the new strategy that my lab is suggesting and the companies that I co-founded, that we can inhibit NO and reverse all of the molecular deficits that I mentioned that, of course, will lead to reversal of synaptic dysfunction and the behavioral ones.
That's fascinating. And I mean, you came obviously from a Shank 3 lab. Is that why you think you just sort of happen to make the connection with nitric oxide, or is there something about Shank 3 in particular that kind of makes the increase in nitric oxide worse, for instance?
Yeah, this is amazing question. So I think the reason we went to Shank 3 wasn't random. When we talk with Dr. Fang, Dr. Tannenbaum and I, we did some investigations on different models. And nitric oxide is produced in the neuronal cells by enzyme named nNOS, neuronal nitric oxide synthase. And this is activated by calcium. So once we looked into different papers of Shank3, we saw that Shank3 mutation leads to calcium imbalance. And once you have calcium imbalance, this will lead to dysregulation of the nNOS. And therefore, we thought that there's a great link between Shank3, calcium, nNOS, and nitric oxide. And this is why we actually very excited about this. And the results of the advanced science paper show that there's an important link between NO and PMS.
Great. So you mentioned using maybe a compound or something to help reverse the ill effects of the increased nitric oxide. Where are you with those studies? Or can we expect anything in humans?
Yeah, so I'm very excited to update that, year ago, we co-founded together with Amir Avniel, the CEO, and together with two Nobel laureates, Professor Shechtman from the Technion and Professor Korenberg from Stanford, we co-founded a company named Neuronos that is based on the findings of my lab, trying to build up this a company in order to develop small molecule to treat PMS. I'm very excited to update the PMS community that we were granted by the FDA Orphan Drug Designation for our small molecule, which is now under intensive and extensive IND enabling studies. So for the people who doesn't know what's IND, it's the studies that we need to do as a company in order to submit to the FDA and to get approval to start first in human. So if you want, I can tell you more about what we are doing right now with the IND and where we are going in the future with the company.
Yeah, if you don't mind to give us kind of a brief summary, because, I think this process, is always good to reiterate to our families because it is such an important part of those clinical trials.
Yeah, so Neuronos now on a stage of um, developing or conducting the IND enabling studying. So we are working with LabCorp and Cambrex, two big US companies that are developing for us the molecule. We are working on oral formulation for the drug, and we are also very soon in the coming months starting toxicological studies. So as the FDA requests, we will inject the drug into rats and dogs and see whether the molecule is toxic or not. And we hopefully that by the end of 2026, we will start first in human, not only for PMS, but also for autism spectrum disorder. So this is actually where we stand right now. Very exciting times.
Yeah.
Yeah, we hope that, we'll get negatives in the tox studies and
Right.
we can go ahead with the clinical trials.
Yeah, this is super exciting. And, I know that sort of thinking the end of 2026 is 18 months from now, which is kind of long, but at the same time, in the clinical trial world, as to remind our community, it takes a long time and a lot of money and a lot of
Yeah.
man and woman power. So, but we're really excited. I know we'll definitely keep updated with you to see how things are progressing. And I'm sure our families will be eager once there is a first in human trial to maybe participate. So we'll keep our community updated.
Yeah, thank you. And again, I'm very happy to be part of this PMS community. Please feel free to the families to reach out. I'm currently in Boston, so it'd be love to meet families and people and get any question that you have. And good luck for all of us.
Yeah, for sure. And hopefully we can get you to come to our family conference next year in July in Colorado. And, families can meet you in person there too.
That would be amazing.
Great. Before I let you go, anything else you want to share with our families so they can get to know you a little bit better or know what else you're working on?
Yeah, so I mean, as I said, I'm now as a visiting professor at Boston Children's. We are trying also in my lab, and I co-founded a company named Point Six Bio that developed diagnostic tool for autism and PMS. So we are using blood samples and machine learning and AI models in order to diagnose autism and PMS through a simple blood testing. This is another efforts that we are doing.
That's exciting. We know that underdiagnosis kind of plagues the whole rare disease community. And so
Yeah.
we're glad to hear you have some
Yeah.
efforts there. And there might even be a podcast about that later that we might need you on too.
Yeah, well, that'd be great. And another thing that I want to mention also on the PMS, so we published a paper on sex differences, and this work that was published in Translational Psychiatry showed that when you use Shankri females and males mice, we don't see any differences. So this is another alert to the community that underdiagnosis of girls and the awareness that there are looks like more girls that have autism and PMS more than we know. So this is another point that I'm happy to mention as well.
Yeah, thank you for mentioning that is very important for our community to know.
Yeah,
And hopefully that can also get out to medical providers, geneticists, everyone
yeah.
out there. So, well, thank you again, Dr. Mall, for being here. We really appreciate it. And we're most excited just about sort of a new avenue for treatment, hopefully. And fingers crossed that we'll talk to you and get some good updates soon.
Thank you so much.
Take
Thanks.
care. Bye.
Bye bye.